Welcome to our Glory Globe TV. Here's breaking news is, did smokers do better than non-smoker in a clinical trial for an experimental cancer treatment? That was the intriguing question that led University of Iowa researchers and their colleagues to develop a drinkable carbon monoxide infused foam that boosted the effectiveness of the therapy known as autophagy inhibition in mice and human cells. Yeah, the findings were recently published in the journal Advanced Science. Looking for ways to exploit biological differences between cancer cells and healthy cells is standard approach for devising new cancer treatments, but it is a painstaking process that requires a deep understanding of complex cancer biology and often a dose of unexpected insight. Researchers have known for several decades that autophagy, which is cell's natural recycling system, is increased in cancer cells relative to healthy cells, suggesting that inhibiting autophagy might be a way to target cancer cells. However, results from almost 20 clinical trials testing autophagy inhibitors have been inconclusive. Within those clinical trials, they found mixed results. There was some benefit, but for many patients, there was no benefit, which really pushed researchers back to the drawing board, says James Bryan, MD, PhD, UI Assistant Professor of Radiation Oncology and Biomedical Engineering and senior author of the new study. Searching for insight into why autophagy inhibition only seems to work some of the time, the researchers made the surprising discovery that smokers in two of the previous trials of autophagy inhibitors seemed to do better than non-smokers. When looked at how the smokers did in these trials, we saw an increase in overall response in smokers that received in autophagy inhibitors compared to non-smoker patients. And we also saw a pretty robust decrease in the target lesion size, Bryan said. This was an exciting finding for Bryan and his team because smoking is also associated with increased levels of carbon monoxide, a gas molecule that can increase autophagy in cells in a way that researchers think might enhance the anti-cancer effects of autophagy inhibitors. We know also that smokers have higher carbon monoxide levels, and while we definitely don't recommend smoking, this suggested that elevated carbon monoxide might improve the effectiveness of autophagy inhibitors. We want to be able to harness that benefit and take it into a therapeutic platform, says Bryan, who also is a member of University of Iowa Holden Comprehensive Cancer Center. Then the report, Glory Globe TV, Delhi.